In lab number 7, we're going to learn how to use redox titration method to quantify vitamin C content in a commercial product. So in this lab, we're trying to analyze a commercial product for vitamin C content via the redox titration. So the product we're going to use is this emergency powder. Right, it's a flavored powder. It claims that it contains 1000 milligrams of vitamin C. Vitamin C actually is ascorbic acid, which has this structure shown in the middle. And the scientist who discovered at the very first was Linus Pauling. Right, so Linus Pauling was the Nobel Prize winner, and he also claimed that vitamin C is used for human body to against cold and the flu. So we can find vitamin C in many of these foods, right? So normally when we're talking about vitamin C, the first food coming in mind is those fruits, right? Those fruits. But actually, a lot of vegetables also contain vitamin C. And in this table, you can see actually some of the vegetables, right, contain a higher vitamin C content than those citric fruits. The quantification of vitamin C is done through redox titration. The idea is very similar to what we have done in the iodine clock lab. So inside the burette, we're gonna fill the potassium iodate solution, right? So this IO3 polyatomic anion is called iodate, right? End with ATE. Under acidic condition, it can oxidize iodide and generate this iodine, right? So this iodine, elemental iodine, and this iodine, right, we can react with the vitamin C or the ascorbic acid, right? So in the second equation, this ascorbic acid is actually the reductant, right? So the iodine generated in the first equation is the oxidant, right? So it's the oxidant. So for iodine to be reduced to iodine, right? So for iodine to be reduced by iodine, and the reductant is the vitamin C, right? So the iodine become iodide, right? So as it is shown in the second equation. And for the vitamin C or the ascorbic acid, as a reductant, it's gonna be oxidized, right? It's gonna be oxidized into this form, right? So if you look at the structure, right, from the ascorbic acid to its oxidized form, it's gonna lose, right? It's gonna lose this 2 protons on the OH attached to this 5 carbon, not the 5 carbon ring, but this ring structure, right? So these 2 protons are being removed. So the idea is that in the first reaction, right, so when this iodine is produced, it will quickly react with this ascorbic acid, and this iodine is quickly consumed, right, so generate this iodide. If all the ascorbic acid is consumed, this iodine produced in the first reaction, right, will start to accumulate, and it will start to bind with starch, right? So we add starch in our system. So at the beginning, when we still have ascorbic acid, the iodine will react with ascorbic acid, right? So when all the ascorbate is consumed, this iodine will accumulate and start react with starch, right? So we know from the iodine clock lab, this complex has this very dark black or dark blue color, right? So in this case, it's gonna be dark black color. So I recorded a couple video, right, when I ran this experiment. For the first video, you can see that when the drop of iodate solution goes into the mixture, you can see this brown colors form right away, but when I shake the flask, the color goes away. This brown color is actually the combination of iodide formed and with the iodine formed and the iodide originally added in the mixture, right? So these two can actually combine together and form this iodine 3 minus, right? So this also will react with the ascorbic acid, right? So the iodine is being consumed right away when I shake the reactor. In the second video, right, so this is when I add this product in the mixture already. So you can see the endpoint actually, right? So you can see the dark color start to form and it doesn't go away. So that's an indication of the endpoint of this titration process. Let's see that again. Right, so notice when I shake it, the color goes away. Right. Now the endpoint is reached, so the color does not go away. To determine the vitamin C concentration in this commercial product, we must know the concentration of this potassium iodate, right? So part 1 of this lab, we're going to determine the concentration of potassium iodate. So these are all the solutions or chemicals we need, right? So we need the starch, we need the potassium iodate, which is to be filled in the burette, and also the standard ascorbic acid solution, because these reactions require acidic condition, so we also need to add the hydrochloric acid. And for the iodide, we're gonna add the solid form, potassium iodide. So first, I pipette 25 milliliter of this standard ascorbic acid solution with a concentration of 0.072 molar. Right, so if I have the concentration and I have the volume, so I can calculate the number of moles of this ascorbic acid added, right, into the flask. Because it requires acidic condition, so I add about 5 milliliter of this hydrochloric acid. Then I measured about 0.5 gram of potassium iodide, right? So this is enough iodide, right, to be oxidized into iodine, right? So this iodide added, as shown in the first equation you see in the previous slide, is in excess, right? So you can barely see the powder in this wave boat, right, because the color is also white. So after I measured out about 0.5 gram of this potassium iodide, I add this solid into this ascorbic acid and containing the hydrochloric acid. Right, so this solid dissolves right away, right, because the potassium salt, right, so all the potassium salt can dissolve in water. Then I add about 20 drops of this starch solution. So the picture in the right, you can see this is the final mixture. Again, it containing this dissolved potassium iodide, right? It also contains this ascorbic acid, and also the starch, right? And also the hydrochloric acid, right? So the cloudiness is due to the starch, okay? Then I fill the burette with this potassium iodate solution, and then I start the titration process. So for the picture in the middle, you can see that's when the endpoint is approaching, right? So the solution starts to get a little bit purplish, right? So that's the color of this intermediate, right? But the endpoint has not been reached yet. And in the picture on the right, you can see the dark color has been formed and that it doesn't fade away, right? So that means the titration has been finished. So I have done these two trials, so the initial and the final volume in both trials are given here, and you need to use this volume in your calculation, right? So put these numbers in the Excel. To calculate the concentration of potassium iodate, let's take another look at these two equations. Right, so in equation 1, right, based on the coefficient, so we have 1 mole of potassium iodate can give you 3 moles of iodine. And in equation 2, each mole of iodine produced is going to react with 1 mole of ascorbic acid. Right, so this is a 1-to-1 ratio. So if you consider the two reactions, right, because this iodine reacted in the second equation is the iodine produced in the first equation. Right, so that means for every 1 mole of potassium iodate, it's gonna consume 3 moles of ascorbic acid, right? Because if you try to match the coefficient of this iodine, right, produced and consumed, right, that's gonna be 1 mole or 3 moles of ascorbic acid, right? So we have this 1-to-3 ratio between the iodate and ascorbic acid. So from our initial and final reading in the burette, we can find the volume consumed, right, for the potassium iodate, right? So this volume consumed is known, right? So on the numerator, we have the concentration of ascorbic acid, which is 0.072 molar, and the volume is the amount we added, which is 25 milliliter. Right, so based on M1 V1 equal to M2 V2. Right, so in this case, we're gonna divide this by 3, right? If this is the ascorbic acid, right? So this side is the iodate, right? So this side is the vitamin C, the ascorbic acid. Let's see, it's the ascorbic acid, right? So here we have the M2V2, right? And they have divided by 3 here. So if we move the M1 to this side, which is the volume of the potassium iodate, right, consumed, we can calculate the molar concentration of the potassium iodate. So in part 2, we can now use the concentration of the potassium iodate, which is calculated from part 1, right, to determine the amount of ascorbic acid contained in this commercial product, right? So when I did this experiment, I opened up this packet. So notice that each packet contains 9.3 gram of this powder, right? So I measured out 0.5 gram of this powder and added in about 100 milliliter of DI water. And so then I add the starch, the hydrochloric acid, and the potassium iodate, right, using the same amount as I did in part 1, right? So pay attention, right, because I only add 0.5 gram, right? So the amount of ascorbic acid I determined through the titration, those ascorbic acid come from 0.5 gram, right? So later I have to calculate how much is contained in 9.3 gram in this whole packet, and compare my result with this labeled concentration, right? So it claims that it contains 1000 milligram of ascorbic acid, right? So let's see how our result, right, compares with this labeled concentration. So this picture here, right, is the mixture of 0.5 gram of this powder, right, with this everything listed. So again, I'm using this video, right? So you've seen this video before. So this is the titration process. When I did this, it took only about 1 milliliter of this potassium iodate solution in the burette. Right, let's see that video again. And notice the starting color was already a little bit orange, right? So that's the color from the powder. So I did this 2 trials again. So we have the initial and the final volume in trial 1 and the trial 2. Right, so using final volume minus initial volume is the amount of potassium iodate consumed. So to determine the amount of the ascorbic acid contained in that 0.5 gram of powder, I use the similar approach again, right? So first, based on the volume of potassium iodate consumed and the concentration I calculated from part 1, so this term is the number of moles, right? So this is the number of moles of potassium iodate consumed, right? So if I multiply this number of moles by 3, that's the number of moles of ascorbic acid consumed, right? Due to that 3:1 ratio, right? So I get the moles of potassium iodate, right? And I multiply this number by 3 to get the moles of ascorbic acid consumed. So that's the number of moles of ascorbic acid. And to convert it into mass, I multiply its molar mass, which is 176.1 gram per mole. Right, so now using this equation, the mass is the mass of ascorbic acid in 0.5 gram. Right, so this is the mass, right? This is the mass per 0.5 gram of this powder. So to find out the mass in the whole packet, right? So after we get the mass in 0.5 gram, we multiply 9.3 divided by 0.5, right? So we can find out the mass in the whole packet, right? So using the data I provided, you're gonna find out that this number is gonna be close to 100 milligram, right? Which is about 1 gram, right? So your result is gonna be in the unit of gram, right? So you're gonna compare result with this 1 gram, right? So to calculate the percent difference. So I have uploaded a Excel template for you guys to work with. Again, for each cell, put the initial and the final volume, and for the rest, you need to show me your calculation, right? So you must use the equation in each cell, right? And to do your calculation. And for part 2, it's the same, right? So the initial and the final volume, I give you those in the slides, right? So for the rest, you have to show me your calculation. So there are 2 post-lab questions, right? First is calculate the percent error, right? Or the percent difference. We shouldn't say error, right? It's the percent difference based on the amount you determined and the amount labeled on this package. And another problem, right? So the second problem is based on a real story happened to me. Okay, so I also drink this at home. So for the report, because all the classes are online right now, and I'm not expect you to be on campus, so everything will be uploaded on Moodle. So I'm gonna need your Excel, right? So complete the Excel, your a photo of your pre-lab, right? So make sure you study the handout, right, before watching the video, right? So take a picture of those things you write down in your notebook, and also this answer for these 2 post-lab questions. You can either handwriting it and take a picture, or you can type your solution in the Word file and upload it on Moodle also.